The recordings will be available on Moodle on the recording tab. Before I get started, this is where I was. So nerve and muscle. Okay. Recording. So remember, I'm not going to cover all of the slides from the PowerPoint from these chapters. There's some of the major ones that gives us the concepts. So concepts that relate to nerve physiology, and then the concepts that relate to muscle physiology. Remember, this is an advanced course, which means I'm expecting you to know some of the basic concepts and terminologies that are covered already in intro physiology courses. But again, if you need to review or if you want me to talk about a few things, you can always contact me and we can go over some of the concepts for bringing you on board. All right. Okay. We have the molecular gradients across the cell membrane. That's something that's really important in terms of concepts, knowing the gradients that will help us with other concepts. Then we have chapter 5 that focuses mostly on these topics. And then a combination of chapters focuses on the muscle. Predominantly, we will be covering skeletal muscle and then compare it with cardiac and smooth muscle at the very end. So if we know a pretty good understanding of skeletal muscle, comparing and contrasting it is much easier. Okay. Again, I'm going to start using the terms ligand, receptors, potential, action potential, graded potential. If you need me to explain those in simple words, please let me, right? Okay. So molecular gradients across cell membrane. Just a quick review here. I'm not expecting you to know the actual numbers from these, just where it is high and what is high, okay? Extracellular fluid, intracellular fluid. And I am an interactive person, so if you can just try to answer my questions, it is more engaging for me, all right? So what does ECF mean to you? What are the compartments that make up ECF? Come on, you can do this.
The fluid outside of the cell?
Very good, outside of the cell. Again, we can go deeper in terms of a lot of things, but outside of the cell, ECF would be plasma and interstitial fluid. Yes or no? Right? That make up the ECF, which essentially means that these are part of the blood plasma. Okay. Then ICF, on the other hand, if ECF is outside the cell, ICF would be inside the cell. Yes or no? Intracellular fluid. Now, I want to highlight a few things here. I want you to focus on sodium, calcium, chloride, glucose. Okay. And then, different color for potassium. Okay. Not that other things are not important, but these are some of the things that we will come across more often. Okay. So what do you see? Why I highlighted those? What's the concept behind? I'm not asking you to memorize 142 milliequivalent per liter, but what is this I'm looking for here? Yes, Lauren.
Potassium is higher inside the cell, whereas everything else is higher outside of the cell.
Perfect. You made my day. Yes. There you go. Okay. Got it, everybody? That's the concept. Okay. And what additional information that you can give me in terms of looking at these molecules, like these sodium plus, K plus, Ca2 plus, Cl minus, what do they also bear? They bear?
Charges.
They're ions.
Very good. They are ions. They have a charge. Yes or no? Right? Again, this is where we bring in the concept of like charges repel, opposite charges attract. So that also is, what do we call that? What Lauren just told us was chemical gradient. Yes or no? Right? What the charge tells us, electrical gradient. Right? And that's why we use the term electrochemical gradient. Correct? That's the word that you get there. Okay, again, I'm not looking away from the camera, I'm just looking at you. I have you on the sponge. Okay, are we good? So you should know the different gradients in terms of chemical versus electrical. Sodium, calcium, chloride, glucose, they are high outside the cell, whereas potassium is high inside the cell when compared to its concentration on the outside. Okay. And one more important information is that chloride bears what charge?
Negative.
Negative charge. That's important. Okay, so negative charge, whereas sodium ions, calcium ions that are outside bear positive charge. All of these information are very got it? Yes? Are we good? So active transport of sodium and potassium. Again, I'm just jumping into this sodium potassium ATPase pump. How many of you are familiar with this? So what does sodium potassium ATPase pump do? They are part of active transport. So the Na plus, they try to go from outside to inside. Okay, because of chemical gradient, yes or no, right? Na is high outside and Na plus is low inside. They try to go inside, and then K plus, what will the status of K plus be? I'm not talking about any other thing, just purely on chemical gradient. I'm not bringing in electrical yet. Just purely on chemical, K plus would want to go out, yes or no? Right? Because K plus is high inside, which means this constant movement can change that's where the term comes in, potential of the cell. And in order to get that potential taken care of, this sodium ATPase, potassium ATPase pump will try to actively put sodium where? This is trying to come in, right? The sodium potassium pump will try to actively put sodium back to where it belongs to. And it will try to put the potassium, actively put it inside to where it belongs to, in the ratio 3:2 to maintain the cell's potential. Okay, got it so far? And when I use the term active, what do I mean? It utilizes
ATP.
ATP, very good. So remember, ATP, the energy from ATP comes only when you break it down. Yes or no? Right, this is where biochemistry comes in. Hydrolysis of ATP releases energy. And for hydrolysis, you need water and an enzyme. What is the enzyme? It's on the slide. What is the enzyme that helps with hydrolysis of ATP? That's part of sodium potassium ATPase pump. Come on, I'm almost getting the answer. I already gave you the answer. Yes. You go by round,
Yes.
right, Mariko? Yes, Malak? ATPase. Very good. It's right here. Yes, that's inbuilt on the pump. Got it? Utilizes ATP. But remember, whenever it's ATP hydrolysis gives us energy. And that energy is required to do this movement of putting Na plus to where it belongs and bring in potassium to where it belongs. Got it? Now my question for you, critical thinking, right? What happens if there is no ATP? Come on. What happens if there is no ATP? You just told me active transport requires
Less.
ATP. Yes, yes, Mala?
You'll have less energy.
Okay, we'll have less energy, which means what process is impaired?
We can't use active transport. Would it revert to passive?
No, the sodium that got inside will be stuck inside, right?
Okay.
Remember, passive, the word passive comes only when the membrane is able to allow these ions to go passively, which is possible only for hydrophobic molecules, right? Or gases like carbon dioxide and oxygen. These are ions. They can just penetrate through these phospholipid bilayer. That is a known concept, right? So these need some helpers, and that's why you have this ATPase pump, okay? So no energy means, obviously, no ATP means no cellular energy, then the sodium will get stuck inside and potassium will get stuck outside, correct? And when you compare sodium and potassium, which is highly osmotic gradient, which has a more osmotic gradient when compared to those two ions. Just take a guess. When you compare sodium and potassium, which has a more osmotic gradient? You have just two options, sodium, potassium. Take a guess.
Sodium.
Sodium. And imagine if it gets stuck inside the cell, what happens to the cell? It will start
Depolarizing.
swelling,
Swelling.
because
yes.
it will bring in what? What comes with sodium?
Water.
I'm just making connections here, guys. What comes? So sodium is stuck. It starts pulling in water. And that's why I use the term osmotic gradient, right? The term osmosis means it's drawing water. So the cells swell. And if we don't, or if the body doesn't address it, it will burst. And that's not a good thing. And that is one of the reasons why ATP is crucial. Yes or no? Okay. And remember, a lot of these cellular processes, physiological mechanisms depend on what? ATP. And that is why med schools require biochemistry. I'm just kidding. Biochemistry is where we learn about ATP. Yes or no? Oxidative phosphorylation, glycolysis. Okay. Everybody good so far? Okay. One of the things that you notice when I do lecture is I try to make connections. Okay, that's more important. Even though we study each and every topic individually, they're all intricately connected, and that's what makes the body amazing, right? So everything is connected, everything is talking. That's really, I mean, I've been teaching this physiology. It's always amazing to talk about these things that our body is doing all the time, right? Whether we eat, starve, stress, happy, fun, movies, binge watching, you name it, the body takes care of us, okay? Until we totally mess it up, okay? All right, so moving on. Simple diffusion of sodium and potassium. I want to introduce the channels here, okay? Again, as much as possible, I will try to focus on the terms, but if you need more background information, please let me know. I can always send you articles to back it up. So there are leak channels. So leak channels allow the movement of ions using their dedicated channels, like, going from outside to inside. So this is a dedicated sodium leak channel. And they are typically open. And this is a dedicated potassium leak channel. And they're typically open. What do you think they would do? What is important for us to know? The gradient, chemical gradient, yes or no? If I have a dedicated potassium channel that's open, the potassium is going to go from inside to outside. If I have a dedicated sodium leak channel open, sodium is going to go from outside to inside. Because we know that sodium is high outside, potassium is high inside, correct? That's the basis. Got it? Okay. All right. So these are the leak channels. Now, remember, when they go through these leak channels, they are going from the region of high to low. High to low. We use the term simple diffusion. This is where passive comes. Right? Passive, does it use energy or not?
No.
No. Thank you. Right? This is where the term passive comes. No use of ATP. Got it? So that's the difference that I want you to understand. Active means Typically, we use the term, we'll go back, active transport, the term pump is one connection. You see the term pump, it is referred to active transport, which means it requires ATP. Remember, when do we use the term pump? When it goes against concentration gradient, yes or no? Which means if I climb the stairs, I might require more energy than climbing down the stairs, yes or no? Right, so you're putting sodium to where it belongs to. It requires energy. Are we clear? So this means low to high. So against concentration gradient means low to high, it's active, pump ATP. High to low is passive, does not require ATP. Got it? Okay. Now moving on, membrane potential, very, very important concept. It's a charge difference. This is where we bring in chemistry tying into physics, the concept of charges and potential, okay? So knowing that K plus and Na plus, they all have charges, isn't it? Right? So these are going to create some charges along the lipid bilayer, okay? Now, how can passive diffusion of potassium and sodium lead to the development of negative membrane potential? So even, K plus is moving from inside to outside, sodium is coming in, sodium also has a positive charge, yes or no? Right? Potassium leaving, the cell is losing positive charge, but it is regaining positive charge. Why would, if you measure the membrane potential of the cell at this stage, it is still showing negative? Any idea why? So the reason being, inside of the cell, so technically the measurement goes like this. We use a voltmeter or galvanometer to measure it. So it is across the membrane. It seems that the DNA, you have those proteins and all that, right? Inside the cell, it has a lot of negatively charged particles. So even with these positive charges going in and out, right? The negative charged particles are going to impart charges. And another reason, another important reason, if we kind of sideline this, let's say, hey, there are going to be negative charged particles. Aside from that, is there a reason why the membrane potential shows negative just purely based on this movement? Are you able to reason out something just based purely on this? It's the gain and loss concept, right? The cell is losing more potassium than gaining sodium. Does it make sense? Okay, just to compare the leak channels without bringing in all the DNA and proteins. That's in addition, right? The cell loses more K plus, which means the leak channels that we talked about, correct? The leak channels for potassium has to be fill in the blank.
Closed. Right?
Closed.
Or inactive, like they're not being.
So remember, leak channels allow K plus to go out, right? If K plus is going out, the cell is losing positive charge, okay? If Na plus is coming in, cell is gaining positive. So purely based on this knowledge, why is the cell exhibiting negative membrane potential? Which means, what does it tell us? That the leak channels for K plus is?
Open.
More. Very good, Eric. Yes, more. Not just more open, they are increased in number too. Yes or no? If they are more than the leak channels for sodium, obviously the cell is not gaining enough positive, it's losing more positive, so it is exhibiting negative membrane potential. Does it make sense? Yes, okay. And I know I will keep saying this often, but I developed love for physiology. I'm basically an engineer, but I developed love for physiology because I love how the body works. Okay. I'm able to conceptualize, I'm able to understand. So the more we understand, I think it's much better to, it retains in our mind. Okay. So keep that in. Are we good in terms of leak channels, guys? Okay. So resting membrane potential. This is so the cell is exhibiting. Potential means a charge difference. The resting means when the cell is at rest. The resting membrane potential is denoted as V suffix M. It is in the range -90 to -70, typically given the unit millivolt. Okay, and we just saw that the resting membrane potential is at the negative value. Yes or no? Right? So it's at the negative value because there are more leak channels for potassium. Everybody with me so far? Okay. Now, why is VM close to EK? So the answer is there for you. The membrane is far more permeable to potassium than Na+. Okay. Remember, the term permeable should not throw you off. The permeable here means that more leak channels or potassium leak channels are, as Eric mentioned, are open. Got it? All right. So there will be instances where we use permeability. That should not hinder your understanding. Because remember, they will the concept will say, or the textbook will say, the potassium is now, and the cell membrane is now permeable to sodium. Sodium just can't go inside because it's an ion, right? What does that mean? There are more sodium channels that allow sodium to come in, is the understanding there. Okay, I don't want you to get the permeability concept differently just because they use it. Okay, now if at this state, so assume that this is the resting state, correct? Now, if the resting state, if there are more potassium channels open at this stage, even more, what do you think will happen to VM? Which direction will that proceed? It's already at negative, yes or no? Right? And if the cell is opening more potassium channels, what do you think will happen? What will happen to VM? Will it become more negative? Right?
More
And
negative.
yes, it will go this way. And it will almost reach the equilibrium potential of the potassium. That's like the very negative. Okay. Now, what happens if at this stage, rest, if I open more sodium? Where is the VM going to go? Remember, if you open sodium channels, sodium is going to come in. Sodium has a positive charge. So what will happen to VM if more sodium channels are open? Which direction will that go?
It will become more positive.
Very good. Thank you so much. Yes, it'll go towards the positive VM. Okay, so every time sodium channels, now we are going to step our foot into gated channels. So far, we have had discussion about leak channels. We're going to step our foot into gated channels. No matter what channels, sodium is going to come in, because of the chemical gradient. Potassium is going to go out. So it is going to have an effect on the VM, correct? Everybody with me? Right. Okay. So again, we have seen few things. I'm hoping that we will be able to get this the next one. So effect of changing permeability of sodium and potassium, just to reiterate, what effect does increasing, I want you to tell me what, this question in simple terms. This is very easy, but they have just made it complicated by using the term permeability. What does this question tell us? Anyone?
VM is becoming more negative.
Very good. I just want you to rephrase that question for me, Eric, if possible, or anyone.
Mm. What is happening if more potassium leak channels are open?
Super. Eric made it. 2 smiley faces. Yes. What happens if more potassium leak channels? What happens to the VM if there are more potassium leak channels? Listen, right? That's how you rephrase this question. Got it, everybody? What happens? The VM goes this way. Okay. And now that Eric told us, I'm sure we can rephrase this question. What happens to VM if there are more sodium ion leak channels? Then it will take it. Super, right? Yes. All right, I'm going to move on unless you stop. So resting and action potential. This is where action potential come in. So resting, we have seen the effects of leak channels. Yes or no? I'm going to use a different color. So leak channels, there are potassium channels, there are sodium channels, and potassium channels are more when compared to sodium. And the leak channels, how are they counteracted? These keep going, right? K plus going out, Na plus coming in. What brings them to a steady state?
The
What-
sodium potassium pump.
Very good, right? So this always works to make sure it comes to a steady state because we need the cell to be at rest all the time, even though these are fluctuating. Got it? So we know the work of this, we know the work of this. Now comes the action. What is action? The cell is getting excited. 1 o'clock, Dr. Bhaskaran is talking, excited, right? Action potential, we need gated channels. What voltage gated channels? So far we have seen leak channels, sodium, potassium, ATPs, pump. Now I'm bringing in what? Voltage gated channel, okay? So this is where you have to understand the cell is going from rest, is becoming, I'm going to use the term action. It's basically excited, right? And then they have to come back to rest. Yes or no? It's a very logical concept, simple. Okay. So rest is something, the stimulus. Are you familiar with the term stimulus? So the stimulus is basically trying to stimulate and make sure it's causing excitable. So let's say we have the resting membrane potential is -70. So some stimulus comes in and gets the VM to -55. So -55 is it getting less negative, guys? -70 to -55. So that is the threshold. So every cell in our body operates at a minimum threshold. So let's say a cell has this as the threshold. So once VM reaches -55, what happens? That will start the depolarization. So depolarization is when the cell is becoming more positive. But here we talk about not the opening of leak channels. Remember, leak channels are always going to be there. Yes or no? Right? They're going to be there. They are opening voltage gated sodium. Yes or no? It is still depolarization, right? More positive. So it has to involve sodium, correct? But what channel are we talking about? Voltage gated. Voltage gated are powerful. They bring in more sodium that it can take it all the way to a very positive voltage. It can go all the way up to +61 millivolts, depending on the size. Got it? And that's where overshoot means positive, closer to much higher positive. And then the cell has to come back, yes or no, right? So this action is the involvement of voltage gated sodium first, that is depolarization, followed by repolarization. So this repolarization is coming back. Color, just so repolarization is coming back towards resting. Now use voltage gated. What do you think is open now? It's repolarization. This is becoming more positive. This is coming towards a negative value, correct? Going back to resting. So voltage gated, what channels?
Potassium.
Very good, potassium. Yes or no? Right? And then because voltage gated potassium channels are less powerful than the sodium ones, these are very, very slow to close. They keep themselves open for a long time, causing hyperpolarization, more negative. That's about it. Okay. But the only problem here when the cell gets stimulated, right, is what? What is the problem? Voltage gated sodium channels, they open, correct, bringing in sodium and close. So where is the sodium stuck? So at the depolarization, the sodium is stuck inside. Correct? Okay. And then the voltage gated potassium channels, they open and they close slowly. Eventually, what happens here? It's stuck outside. Got it? So this also very similar to what happens during the leak channels. Yes or no? They keep going out, out, in. So what comes to the rescue? Our favorite pump. Yes or no? Right? Plus leak channels. Hey, let's come to the rescue. We will try to make it up. Okay? So this is something that's very important. Right? Depolarization, hyperpolarization. So when I say more negative, I can use the term hyperpolarization. If I say more positive, I can use the term depolarization. If I say action potential, what is the quick connection in terms of gated channel? Voltage gated sodium channel. Makes sense. Okay. Depolarization is going upward. Repolarization, note the term RE, going downward. Okay. Are we clear? So please become familiar with these. Action potential characteristics. We'll just, in 2 minutes, we'll go for a short break. So an action potential is regenerating depolarization. So we all know that it propagates without decrement, is excitable. So these are the characteristics. All are non-event. What does that mean? Only if it is reaching the threshold, an action potential will be generated. No threshold, no action potential. So it's all or none. Got it? It's constant amplitude. So if there is a constant stimulus, what happens to the APs? Do they go this? They're all the same amplitude. If it's frequent, what happens? The frequency increases, the number of APs, they come together closer, but their amplitude, their tall, how tall the waves are, that remains constant. And that's what amplitude means. Am I clear? Okay. So these are some of the characteristics. Depolarization initiated by exciting stimulus involves in changes in permeability. What does that mean? Sodium coming in, correct? That is what permeability means. And they rely on voltage gate. Now you understand why the importance of voltage gated ion channels are. So this is upward stroke, downward stroke, depolarizing and repolarizing. This ENA refers for, and then EK for repolarizing. And EK is standing for towards potassium permeability. All right. Everything comes down to the basic understanding. And then we have, okay, we just ended with this one for our break. So refractory periods. So we have absolute and relative. Absolute is when an action potential is currently happening. When it's currently happening, there is absolutely no room for another action potential. Okay. So voltage gated sodium channels, there's a lot of information about the different channels. If you take a closer look at those channels molecularly at the cellular level, they have something called inactivation gates. So they are inactivated. So they don't allow another action potential at this absolute period. But when the cell is repolarizing, the relative refractory period starts. So this is more on the potassium channel. And when the cell has a greater stimulus, there might be a chance of an action potential. Okay, so absolute refractory period, currently an action potential is happening, there's no room for an additional one. Relative refractory period, it depends on the stimulus. There could be an action potential during that period. Got it? Okay. So why refractory periods are important? This is especially important in cardiac muscle. It allows the cell to completely, get activated, and they limit the maximum frequency of beats. This is especially important in cardiac muscle. Got it? Okay. We'll take a 5-minute break and then resume with our Zoom session. I hope you will be joining. All right. I'm just going to point out a few things. If we need more in-depth discussion, I will go in. So myelination is providing insulation. The Schwann cells in the peripheral nervous system helps with myelination. So the myelin sheath is actually white. It gives the color white. And it surrounds the axon. You see here, it surrounds the axon. It provides insulation and it helps with increase in speed of transmission. Okay. And then this is how it's laid out in the peripheral nervous system. So once we have a lot of information coming from the nervous system, we will get into myelin and it. Okay. So when we talk about multiple sclerosis, remember, myelin provides insulation and increased speed. In the CNS, it's actually made by oligodendrocytes. So if there is a problem producing myelin in the CNS, okay, it is given a special name called multiple sclerosis. It's important for us to know. It is an autoimmune or immune-mediated inflammatory demyelinating disease of the CNS, which means the oligodendrocytes are inflamed or irritated. They are not able to make enough myelin. Got it? That reduces the speed of transmission. So what happens? So there is less speed. There's going to be weakness, right? Because everything is talking to our muscles, right? The nerves are talking to the muscles. Everything becomes weak. Eventually, there's going to be neurologic deficits and everything happening. So MS is not easy to live with. Okay, got
I
it?
have a question.
Yes.
How does it get demyelinated?
So this is immune mediated. We have autoantibodies attacking the myelin sheath, right? Or the oligodendrocytes. It is autoimmune. And we have those immune cells are targeted and they are activated because it could be because of inflammation that triggers these cells, right? And autoimmune conditions can also trigger these conditions. Okay.
Thank you.
Then neurons communicate via synapses. So you have point of communication between neurons is called a synapse. So I'm sure you have a pretty good understanding about the neurons, yes? The cell body, the nucleus, the axon, correct? The cell body, and then the axon. Axon is typically surrounded by myelin. So this is a myelinated neuron. Then the cell body has these dendrites. So the dendrites form connections, right? Synapses with another axon, correct? And the synapses typically predominantly involve what? Neurotransmitters. Abbreviated as NTs. There are a variety of neurotransmitters. These are chemicals that are released from the axon. So let's say you have the axon. Then you have this dendrite, right? And this is an axon. Then you have the dendrites. You see this? This is the connection. So this gets going, and this is the synapse. And remember, what do you see on these? This is actually, dendrite is basically nothing but a cell membrane of a neuron, correct? The cell membrane will have what? Ion channels, correct? Just like leak channels, voltage gated channels, and all of those, okay? But the dendrites typically have another type of channels called ligand gated channel. This is where I'm bringing in another term. Okay, so we have heard of leak channels, we have heard of voltage gated, yes or no? Right? This is ligand gated channels. So ligand gated channels are predominantly found where? On the dendritic portion of the neuron. And the axonal region is where you find what? Voltage gated ion channels. So voltage gated ion channels, what comes to your mind? What's a quick connection?
Sodium.
Yep,
Voltage gated channel.
action potential or depolarization, yes or no? Right? If it is a sodium voltage gated channel, then it's going to cause action potential. Ligand gated, any takers on what potential would that be called as? Starts with the term, letter G. Oh, I know that. It's just, it's become history now. Graded potential, okay? Yes, graded, local, electrotonic, all of those are different names. Okay, so graded potential, it's like, you know, all like dendrites look like this. So they are receiving what? They're receiving the stimulus from another axon, yes or no? The stimulus has to be received. The stimulus can be excitatory or inhibitory, correct? If it's excitatory, it is going to bring in positive charges. If it is inhibitory, it's going to bring in negative charges. Or make the cell close to negative. Got it? So it is going to cause a local change first. If that local change, if VM now slowly becomes closer to threshold, so going past the dendrite, they're all activating. If it's touching the threshold, what channels would be activated? Voltage gated, sodium channels will be activated. And that is why dendrites do not have voltage gated channels. Got it? Make sense, guys? So excitatory versus inhibitory. So graded potential or electrotonic potentials, these are basically, and again, all of these are talking about the stimulus. What is the trigger? And the trigger, let's say I have, this is the synapse. We call this neuron as postsynaptic neuron. And the neuron prior to the synapse will be presynaptic neuron, isn't it? Presynaptic, postsynaptic. So this is a neuron before the synapse, this is the neuron after the synapse. I can have this neuron causing positive stimulus. I can have this neuron causing negative stimulus, correct? I can have many neurons like this targeting the same synapse. Which one? Right? So many neurons to one neuron. Okay, so there can be multiple various factors that initiate the stimulus and that causes this local potential. So remember, graded potential is opposite to action potential. Anything that we know about action potential, the graded potential will be opposite, which means they are graded. They don't have refractoriness, they summate, they can have a bigger response, their amplitude can change, right? What about action potential? They're all same amplitude, correct? So whatever we know about action potential, the graded potential is going to be the opposite because it is depending on the stimulus fluctuation, correct? Okay, so just going over this, I'm just going to point out this. Okay, now this is EPSP. What does EPSP stand for? Excitatory postsynaptic potential. Okay, if let's say this is a threshold, correct? Yes or no? If it is meeting the threshold, then there is going to be action potential. No threshold, no action potential. All or none. Yes or no? Right? So when does it meet the threshold? What is required to make the VM go close to the threshold? Graded potential. So what is this trying to do? What is EPSP trying to do? This is at -70 rest. Are you guys with me? This is -70 is at rest. What is this EPSP trying to do? It is trying to bring the VM closer to, come on,
Threshold.
closer to threshold. So that what can happen after that? If the VM is going closer to threshold, what can happen after that?
Action potential.
Very good, an action potential. Got it? Okay, that is why it is excitatory postsynaptic potential. Got it? All right, so this is temporal versus spatial. This is temporal meaning like, one neuron trying to exert a lot of EPSPs. Spatial meaning many neurons are trying to exert EPSPs on this. Got it? It's just the intensity coming from one with more intensity or many to one. That's the difference. But everything is trying to do what? Trying to make it excited. Got it? Okay.
I have a
Yes.
question.
Yeah.
For the spatial summation, does it improve the time it takes? Does it make it faster?
Yes. Because
Oh, okay.
if they all have kind of equal intensity, then it's going to reach the threshold at a much faster rate. Yes.
Thank you.
And what is the inhibitory? If excitatory means it is trying to move closer to threshold, what is it trying to do here? Right? You have resting. Is it moving away from threshold? Am I clear, everybody? Okay. So if it is moving away from threshold, is an action potential likely or not likely? Not likely. Okay, so basically, inhibitory synapses will cause it to hyperpolarization, make it more negative because it is going away from threshold lower than B. Are we clear, guys? So if I have a question, let's say EPSP, is it a graded or an action potential? It is
Graded.
graded. Is it excitatory or inhibitory?
Excitatory.
Great. And then is it getting the VM close to threshold? Is it trying to bring it close to threshold? Yes. Okay. And in terms of permeability, real quick connection. Remember the same thing that we talked about. EPSP, what kind of channels can help achieve EPSP? Make my day here. We're almost there. I want you to try to be very specific. EPSPs are generated on dendrites, yes or no? And I gave you all the information that you can use to give me a specific answer. I want in terms of either channels or permeability. You can use any channel, any ion. Yes, ma'am.
Sodium voltage gated channels.
One more time, you are close with
Sodium
sodium.
voltage.
Dendrite,
The
dendrites.
voltage one. Those are the ligand ones?
Yes, very good.
Oh,
Yes.
so the actual,
Remember,
the
the cell is at
axodendritic-
rest, right? Cell is at rest. The EPSP is trying to bring it close to threshold, which means it is trying to bring in little bit of sodium in, yes or no?
right, okay.
That will make it go from -70 to what? I'm just giving you these numbers if you don't have to memorize. This is just an understanding. It is going from -70 to -55. If this is the threshold, then what would happen?
It'll induce an action potential.
Then it will open voltage gated sodium channels that are found on the axon. Yes or no?
Right,
And because it is also surrounded by myelin and all of that fun things, it will have the action potential. So it is like a logical flow there. Got it, everybody? Yes. Okay. And that is one of the reasons why I keep insisting on knowing the different types of channels. So far, so good? Yeah. So ligands and receptors. So what could be a ligand? So here, I'm just going to, this is an axon ending terminal, and this is the dendrites of another neuron. And these are the chemicals that are released. And these are the receptors for these. What are these called? In general, neurotransmitters. Yes or no? Right? So they are released in this, what is this? Synapse, correct? And these neurotransmitters are also called as ligands. So ligand is a general name given to the neurotransmitters. What do? They get released into the synapse, right? And they look for their cute little receptors. Where? On the dendrites, yes or no? Okay, and these receptors, can receptors be ion channels? Any biochemistry or introphysiology concept? Can a receptor take a form of an ion channel? Yes, thanks, Eric. Yep. So a receptor can be an ion channel. We call that as a ligand-gated ion channel. And I can also have a regular receptor that need not be an ion channel, but that can still bind to the ligand, right? I can have a regular receptor. I can have a channel receptor. This is how a channel would look like. You see the difference? A regular receptor versus a channel. So this is a ligand gated. It looks like a channel. And how it is activated though? It is activated only upon ligand binding to it. What is a ligand? Neurotransmitter. Got it? So the receptors are waiting on the dendrites. When the neurotransmitters are released, they find their own specific receptor, bind to it, and make necessary changes to the VM of the postsynaptic loop. Got it? So we have all of these are different ligands. Fun, ACH, norepinephrine, epinephrine. I'm sure these should ring a bell. So ligand is a substance that binds to a receptor. Receptors are largely named after the ligand. So we have receptors. We'll go deeper into this, but ACH will bind to ACH receptors. You see that? They have their own specific receptors. The receptors can be of multiple types. It can be nicotinic or it can be muscarinic. Okay, so we will go into the depths of this later, but right now our goal is to get familiar with these terms, ligands and receptors. Got it? Okay. So excitatory and inhibitory synapses, important. The inhibitory ones are GABA and glycine. You see the chloride here, guys? Did I mention about chloride connected to GABA and glycine? So what does that mean? Permeable to anions, like chloride means chloride is coming in or going out?
In.
Very good, coming in. Why? Because chloride concentrations are,
Negative.
it's a negative charge, it is high in these. Both needs to be known. Correct? Okay. And if it's becoming more negative, what is it trying to do? To the postsynaptic cell, it becomes hyperpolarization. Yes or no? The term hyperpolarization means more negative. It depresses excitability. Okay. On the other hand, what is excitatory? It's going to do the opposite, right? It's going to depolarize, it's going to enhance excitability. So these are acetylcholine on a specific receptor, and then the glutamate are excitatory neurotransmitters. In terms of these, remember excitation, sodium, open, potassium, close, calcium, anyone?
Open
Why?
to allow it into the cell.
Because calcium is high in these.
Permeability. Yep.
Got it? All right. We are just building on our knowledge, guys. Nothing is, it's really important. Are we good? So inhibitory means going away. Excitatory means going towards. Okay. So next we move on to our favorite second concept about muscle. Okay. So far so good with the nerves, the neuronal aspect of it. Okay. So moving on to muscle, it is important for us to know that muscle, we have three types: skeletal, cardiac, and smooth. The skeletal muscle is profoundly studied because of its various fascinating abilities, right? So again, I will go over these slides, but I will try to give you a quick review of everything put together, okay? So we have, when we cut open the skin and try to look at the muscle, right? When we actually go deeper and deeper, we have these elements called myofilaments. We heard of that? So myofilaments such as myosin and actin. So these are the actual contractile fibers. So basically, when myosin binds with actin, it causes contraction. But the problem is these are found deep inside the muscle. So basically, I have to go layer after layer. Have you heard of the term fascia, right? So we have to basically go deeper and deeper to find these elements. And for these filaments to actually cause this, the skeletal muscle needs excitation. What does that mean? So for the myosin to bind with actin, that's called contraction, right? Excitation is the first step. Without excitation, there is no contraction. Can you please understand that? Okay. The muscles cannot contract without stimulation. And that is why when there is a nerve damage, what happens to the muscle? Paralysis. Yes or no? Why is nerve damage causing paralysis? Because they have to talk. If their connection is cut, the muscles will not have the excitation. No excitation, no contraction. How many of you have heard of this? Excitation, contraction coupling, right? So that's where it comes from, okay? So now we are going to talk about this portion first, and then talk about the excitation later, okay? Remember, this is happening where you have the axonal terminal, correct? That's part of the neuron. This is what gives me. What does it give me? Excitation. Yes or no? Correct? What do I put here? Do I put another neuron or what goes here? Because we're talking about muscle, what goes here? Yes or no? Right? What is this? Neuromuscular junction. So instead of calling me that as a synapse, synapse is usually given the name between two neurons, isn't it? So this is neuronal muscle connection. So neuromuscular junction or myoneural connection. Okay, so we are going to target the skeletal muscle. It has the myosin, then we have the actin filaments. So myosin has these sites that help with binding to actin. Okay. And then the mechanism of muscle contraction for here, the theory here is that calcium is very much needed for muscle contraction. Okay. And the actin, we have the thin filament. The thin filament has actin, troponin, and tropomyosin. So calcium, once it comes into the muscle cell, it binds with troponin and moves the tropomyosin away. So calcium comes in, the muscle gets activated. So what happens? Calcium binds with troponin and moves the tropomyosin away. So that it helps, this process helps myosin bind with actin. Got it? Okay, so this is important. So walk along theory. Remember, the myosin can bind with actin only if it is energized. And where is that energy coming from? Listen, ATP hydrolysis, muscle contraction requires ATP. So if ATP releases energy, then it becomes energized. Got it? So we saw calcium requirement. Now we saw ATP requirement. So both are crucial factors. Lack of ATP is a problem. Lack of calcium is a problem. Okay. Yes, Eric, the release of ACH is where excitation is. I'm not there yet. I'm going to talk about this in a few minutes. Thanks, Eric, for bringing that up. Okay. So excitation, yes, ACH is right here. And that requires voltage-gated calcium channels on the axon terminal. That's part of the excitation. I'm talking about the process that happens inside the cell first. Okay. Does that help, Eric? Okay. So here, I'm just going to go over a couple more slides and then we can kind of wind up the concept in the next class. But when you see here, the myosin head attached to actin, so this is from the last cycle. So we know that myosin binding to actin causes contraction. Yes or no? Right? So this has happened. Now, to continue with another round, what is required? ATP. Yes or no? Right? So ATP is required. There is two things that I want you to pay attention. ATP has two consecutive steps. Number one, it helps first to detach. Remember, this is bound like this, myosin and actin. The ATP comes quickly to detach it. And then what does it do to the myosin head? It helps with re-energizing it. Got it? So please understand, the first job of ATP is to detach myosin from actin. And then energize it for another round. Make sense, guys? provided what else is available? Calcium is still high. Correct? Two requirements, calcium and ATP. So ATP, the myosin head releases, then hydrolysis, do you see that? This is detachment, this is energize, correct? Two consecutive steps. And now the hydrolysis releases energy, and this energy will cause what we call a power stroke. Power stroke meaning myosin binding to actin. Okay, it's a very simple process. You just have to make sure that it helps with these requirements, ATP and calcium. I have posted the animation, but I'm going to play that animation real quick in the next class just so that we have some visualization. So animation helps visualize the mechanisms. So please go over whenever you find time, okay? So I'm done with today's concepts discussion. So if you have any questions, please reach out to me. I will keep discussing few topics and concepts in our Zoom sessions and review them. So let me know if you have any feedback on what topics you want me to go over, or if any questions, please reach out to me. All right, so thank you so much for joining today. I hope to see some students during tutoring sessions or anytime during my office hours. But if any of those doesn't work, please let me know and I will be willing to help you out, figure out a time that works for both of us. Okay, thank you so much. And I hope you have a good rest of your day.
Thank you, Doctor.
Yep.